All right, so with that, Devon, please take it away and I will go dark.
You're muted. You're on mute.
Sorry about that. Can everybody hear me now?
Yes.
All right, perfect. Sorry about that. So coral reefs.
Hey, Devon, can you speak a little bit louder? You're kind of muffled.
Kind of muffled, okay. How's it working now? Is that better?
It's still kind of fuzzy.
Okay, let me see. Okay, how about now? Is that better?
A little bit.
A little bit? Okay.
Yeah, it's closer to you. I can hear it better now.
Okay, better now? Okay. All right, sorry about that. So coral reefs provide many goods and services to us to provide an essential habitat for many organisms in the marine environment. They give us food for subsistence. There's also some economic benefits that are associated with coral reefs, such as tourism. They also give us many recreation opportunities and also coastal protection. However, coral reefs are impacted by multiple stressors. So there are natural stressors to reefs, things such as storms, extreme tides, and anthropogenic stressors, things that we humans are causing, such as pollution and overfishing. And it's important to note that it's not just one type of stressor or just one stressor acting on a reef. It's multiple stressors, a mixture of natural stressors and anthropogenic stressors that are acting synergistically to degrade reefs. It's also important to note that there are land-based stressors, things from land that are stressing reefs. And this is an issue particularly in developed coastlines, and oftentimes development along the coastline amplifies these stressors. One such example of that is sewage pollution. Sewage makes up a large component of coastal pollution. Sewage contains pathogens, synthetic compounds, and other chemicals. Separately, each of these components are harmful to the environment, but when they're together, acting synergistically, their effects are not fully known. So where does this sewage come from? There are two main sources of sewage pollution in the marine environment. The first are on-site sewage disposal systems, or OSDS. These are cesspools, septic tanks, and aerobic treatment units. They offer varying degrees of sewage treatment. Cesspools, for example, are basically just pits in the ground where sewage is put into. There's no treatment going on whatsoever, whereas aerobic treatment units at least offer some nutrient removal before the sewage is discharged. There are also sewage treatment plants, and these are wastewater injection wells where treated sewage is injected into the ground and outfalls where treated sewage is discharged into the environment. But sewage pollution impacts coral reefs. Sewage has been identified as a driver of reef degradation, and sewage is a global issue. This image here is a map of the world's coral reefs by Ware and Vega Thurber in 2015. And in blue, that shows the distribution of the world's coral reefs. And all the red dots show coral reefs that are impacted by sewage pollution. So we can see that it's something that occurs all around the world. It's not really confined to one specific area. Nutrients in sewage can promote algal growth and can actually impair coral health. And pathogens in sewage have been responsible for disease on the reef. One example of that pictured here is the white pox disease. This is a coral disease that is occurring in the Caribbean and has been responsible for the loss of corals there. And the pathogen that's responsible for this disease has actually been found in human sewage. But I also wanted to talk about something that influences stresses on the reef. And one of those influences is water motion. Water motion is a bit of a double-edged sword. It can introduce stressors to a reef, but it can also reduce the residence time and exposure to stressors. One example of that is shown in this diagram here by Storlazzi et al. In 2014. And this was from a study that was done on the island of Molokai. And this diagram basically shows changes in tide and wave motion. And what the changes in tide and wave motion are doing are stirring up and moving sediment, shown as these particles, over the reef. So as I said, it's kind of a double-edged sword. It can introduce stressors, but it can also reduce the stressors on a reef. Water motion has also been associated with coral health. Studies have found a higher prevalence of coral disease on poorly circulated reefs, whereas vice versa, reefs that have a higher circulation generally have higher coral health. But sewage pollution is an issue in particular in the state of Hawaii. There are about 110,000 OSDS units throughout the state, and collectively they discharge 263 million liters of effluent per day. And in addition to that, many of these OSDS units are located near the coastline. The 5 major Hawaiian islands here are mapped by Whittier and Alcati, and this map shows the density of OSDS units on each island. The warmer colors, such as the reds, orange, and yellows, indicate a higher density of OSDS units. And one thing to notice that on all 5 major islands, there are high densities of OSDS units near the shoreline on every major island. In addition to that, on the island of Maui, the West Maui wastewater injection wells have been an issue in recent years. They have been associated with the eutrophication and algal blooms on West Maui's reefs. And as a result of that, there's been a lawsuit that was filed a few years ago, and it's actually been taken to the Supreme Court with a Supreme Court ruling pending. So there's been a lot of time, money, and effort tied up into the West Maui wastewater injection wells. But sewage pollution is a really big issue here on Hawai'i Island. Hawai'i Island has about 200,000 residents here, and about a quarter of them live within a mile of the coast. In addition to that, Hawai'i Island has the majority of the state's OSDS, just over half of the state's OSDS units. This is the same map by Whittier and Alcati, showing the density of OSDS units. And not only do we see that, again, there are high densities of OSDS units near the coastline, but it's all around the island, and on the north side of the island, the east side of the island, the south side of the island, and the west side of the island. In addition to that, there have been few pollution studies conducted here on Hawai'i Island. Many of the pollution studies have been conducted on the island of Oahu or Maui. And even further than that, there have been fewer studies here on Hawai'i Island that have pertained to sewage pollution on coral reefs. Sewage pollution in particular is an issue in South Kohala. Many studies conducted in South Kohala have found evidence of sewage polluted water at Puako's shoreline. Puako is a community in South Kohala. In addition to that, studies have also shown that this sewage polluted groundwater is actually from OSDS units. There's also been some evidence of sewage on the reef at Puako, as well as decreasing coral cover and health in South Kohala. One example of that is this graph from the Hawaii Department of Aquatic Resources, and it's showing a result of benthic surveys, comparing benthic surveys that were conducted in the late 70s and the mid 2000s. We can see that comparing the late 70s to the mid 2000s, coral cover has decreased while turf and macroalgae cover has increased substantially. So my thesis project had 3 research objectives. The first was to identify sewage pollution hotspots at a larger spatial scale. Many of the previous studies in South Kohala have focused around the Puako region, and we wanted to expand geographically beyond that, and also look at regions that use other forms of sewage treatment. We also wanted to determine how water motion influences sewage pollution. There isn't very much, if anything, in literature about this. And as I mentioned previously, water motion can kind of be a double-edged sword. It can reduce stressors on a reef, but it can also bring stressors to a reef. And we wanted to see how water motion plays into sewage pollution on a coral reef. And finally, we wanted to establish if sewage pollution is contributing to impaired coral health. To do this, we use Benthic offshore stations throughout the South Kohala coastline. Many of these stations are also used by the Nature Conservancy just so we can kind of build on some of the existing data and build a long-term record of changes on South Kohala's reefs. We had 20 Benthic stations, but we broke them down into 5 study regions. And these regions were named based on their proximity to luxury resorts in the South Kohala region or the Puako community. So down here in the orange, we have the South Resorts regions. These are located near the Mauna Lani and Fairmont Orchid Resort. We have the Puako community here, and we have the South Puako, Middle Puako, and North Puako regions here. And finally, up north here, we have the North Resorts regions, which are located near the Mauna Kea and Hapuna Beach Resort. At all 20 stations, we collected benthic water samples. We collected these samples as close to the benthos as we could, and we analyzed them for 2 types of fecal indicator bacteria, or FIBs. These are Enterococcus species and Clostridium perfringens. They are bacteria that are associated with human sewage and are also monitored by the Hawaii Department of Health. We also looked at the nutrient content of these benthic water samples, as well as the salinity of these water samples. And we wanted to look at salinity because salinity is one indicator of groundwater, or GW. And as I mentioned previously, studies that were conducted in Puako have found evidence of sewage polluted groundwater. We also collected macroalgae samples from all 20 stations, and we looked at the nitrogen-15 isotope. So the nitrogen-15 isotope is used to identify nitrogen sources, and we can do that using delta 15N, which are basically values of the nitrogen-15 isotope. To look at water motion, we use 2 methods. We use Clod cards, which are hardened plaster blocks that dissolve under increasing water motion, and they kind of give us an idea of the relative water motion in an area. And we attach these directly to the benthic substrate. We also use an instrument called an acoustic Doppler current profiler, or ADCP. This is an instrument that uses the Doppler effect to measure the direction and velocity of particles in the water column. So along with water velocity, we can also look at the depth of the water and the temperature as well. And it took these measurements about 1 meter above the substrate. So we deployed Clod cards at all 20 stations. There are 180 Clod cards in total that were deployed. However, we only had 1 ADCP, so we deployed it at 1 station in each region. And those stations are identified by the triangles here. So we have 1 in the South Resort, 1 here in South Puako, 1 here in Middle Puako, 1 here in North Puako, and 1 here in the North Resorts region. We also conducted benthic surveys at all 20 stations. We looked at the percentage of benthic cover. We looked for coral, crustose coralline algae, or CCA, turf algae, and macroalgae. We also looked at coral health at all 20 stations. We looked for the conditions algal overgrowth, bleaching, discoloration, growth anomalies, and tissue loss. So moving to our results, we found evidence of groundwater present on the reef. What I'm showing here is benthic salinity values on the y-axis and our 5 study regions on the x-axis. And this dashed line here indicates a salinity of 35. 35 is an important number because full strength seawater is typically around salinity of 35. What we can see here is that our salinity Sorry about that. I think I might have got muted there. But what we can see here is that the benthic salinity values for all 5 regions fall below 35. So this indicates that there's some fresh groundwater present on the reef. These are the results of our benthic fecal indicator bacteria concentrations, and both types of fecal indicator bacteria fell below Hawaiʻi Department of Health standards. The dashed line here on the top, we're looking at Enterococcus species. This dashed line indicates a Hawaiʻi Department of Health standard of 130, and our benthic water samples had FIB or Enterococcus counts that were orders of magnitude below this standard. And it was pretty much the same story for Clostridium perfringens. The Hawaiʻi Department of Health standard for Clostridium perfringens is 5, and once again, our samples fell well below that. The next slide here shows the results of our algal delta N15 values, and they represent a mixture of N sources. There's kind of a lot going on in this graph, so I'll break it down. On the y-axis here, we have the delta N15 values. On the x-axis, we have our regions, and these colored horizontal bars here are different nitrogen sources, and the bars, the width of the bars, or the height of the bars, excuse me, is the range of delta N15 values that are associated with that nitrogen source. So on the top here, we have OSGS units from the Pu'oko community, low elevation groundwater, high elevation groundwater, ocean water, and fertilizer. And when we plotted our delta N15 values from algae we collected on the reef, they kind of fell between ocean water and sometimes groundwater or even below that. And one of the things to note is that macroalgae are opportunistic. Basically, they opportunistically uptake nitrogen from any source they can. They don't really discriminate. They don't really have a favorite nitrogen source. They uptake nitrogen from whoever they can, whenever they can. So this might kind of explain why we see a mixture of nitrogen sources. The next graph here shows the sewage pollution scores for the 5 study regions. The sewage pollution scoring method is a method that was developed by one of Tracy's previous graduate students. And it came out of this need to really develop a better way to assess sewage pollution in the marine environment. Traditionally, sewage pollution, you determine if sewage pollution is occurring by looking at nutrient, fecal indicator bacteria, and delta N15 values. But many times, these 3 indicators don't agree with each other. You can have really low bacteria counts and really high N15 values and so on. So this traditional method often provides conflicting results. So what they did was they developed the sewage pollution scoring method that takes all 3 indicators into account, but weighs them accordingly based on their likelihood of sewage indication. And the sewage pollution scoring method has 3 categories: high, medium, and low. I've shaded in the bars for the range of values for the high, medium, low categories. So similar to the N15 graph, we have our 5 study regions plotted here, along with the range of values, sewage pollution score values for each region. We did an ANOVA and found some statistical differences among regions, but what's really important to note is that the sewage pollution scores were highest in the North Resorts region and the South Pu'oko region. Their scores did fall within the high category a few times. And this to us indicates that sewage pollution is likely present on these reefs. The next graph here is based off of our ADCP data, and it's showing how tides expose the reefs to sewage. This is another kind of intensive graph, so I'm gonna kind of break it down for you. On the left side, your left y-axis here, we have water depth. And one thing to note is that I flipped the axes. So these axes go from low values on the top here to high on the bottom. Same for down here, low on the top, high on the bottom. And on the right side, we have benthic water temperatures, and it's the same low values on the top and high on the bottom. And I did this just because it kind of gives you almost a shot of the water column itself. On the x-axis here, we have time. And this, what I'm about to explain occurred multiple times over both study years in many study regions, but I've just picked two regions here to show as examples. So we have two colored lines here. The gray line indicates depth, which changes gradually over time. This is tide changing. We're going into a low tide here, moving up to a high tide, vice versa. And the black line here indicates temperature. And one of the things that when you look at the two plotted together, you notice that, as we see here, there are these rapid changes in temperature, drops in temperature, I should add, that occur either before, during, or after changes in tides. And what we notice is that along with the water motion, this to us indicates that there's some fresh groundwater with lower temperatures. Temperature is another way to identify groundwater. It's often cooler than seawater. That this low temperature groundwater from near the shoreline, once again, to reiterate, previous studies conducted in Puakoa found sewage polluted groundwater near the shoreline is moving over the reef. So this tells us that these tides are moving sewage polluted groundwater influenced shoreline waters over the reef and are therefore exposing the reef to sewage. This next slide here shows the results of our benthic surveys. This is showing the percent cover of coral, crustose coralline algae, and turf algae. And one thing to note is that although we did see some significant differences among regions, turf is the dominant benthic substrate at Puakoa, and this has been identified in previous studies as well. And it's especially dominant in the North Resorts region. That's where turf algal cover was the highest, as well as the South Puakoa region. Also, notably, coral cover varied, was different. Although it didn't statistically vary, it was different, slightly different among the regions, and it was particularly low in the North Resorts region. This next slide shows the results of our coral health survey. One thing to note is that we noticed impaired coral health in every region. This top graph here shows the percent prevalence of coral health. Prevalence refers to the percentage of coral colonies we surveyed. So how, in a sense, how common each type of coral health or impaired coral health condition was. And we noticed that algal overgrowth was the most prevalent condition in South Kohala. Second to the second and third most prevalent conditions are growth anomalies and bleaching. On the bottom here, we have the percent severity, and severity refers to the percentage of an individual colony. So what percentage of the colony was actually affected by this health condition. The most common or the highest severity condition was bleaching, and that was followed by discoloration and growth anomalies. Just to kind of give you a little visual aid, this is what algal overgrowth looks like. It's turf algae growing in live coral tissue. This is bleaching, when corals expel their symbiotic algae. This symbiotic algae basically provides them energy to grow. And growth anomalies, which are these protuberant growths on coral. Speaking of growth anomalies, we found an association between growth anomalies and groundwater. This graph here shows the results of a mixed effects models we ran on the percent severity of growth anomalies and silica concentration. Mixed effects models are similar to linear regressions. They look for associations between variables. But the one special thing about mixed effects models is that they allow for nested data. And this happened when we created our 5 study regions. What we did was we grouped 3 to 5 stations within a region, and we nested them in that region. So essentially, the mixed effects models takes that into account when it does its analysis. And the results of our mixed effect model show in most regions a positive association between the percent severity of growth anomalies and silica. Now, another way to identify groundwater in Hawaii is silica. As groundwater moves through the aquifer, it accumulates silica from the lava basalt. So silica, in a sense, is kind of another way to identify groundwater. And as I mentioned previously, studies in South Kohala have found sewage polluted groundwater. So it's likely that the severity of GAs is associated with sewage pollution. So to conclude, and also to reference my 3 study objectives, we identified sewage pollution hotspots at a larger spatial scale. We confirmed existing hotspots. South Kohala, South Puako is one of those existing hotspots. And we also identified a new hotspot, the North Resorts region. We also did find some evidence of sewage on the reef, but it appears to be diluted and mixed with ocean water and groundwater. We also determined how water motion influenced sewage pollution. In this case, we found that tidal changes are exposing the reef to sewage pollution. And we also did establish a connection between sewage pollution and impaired coral health. In this case, we found an association between growth anomalies and groundwater. Some of the implications of this study are the use of the sewage pollution score. Previously, the sewage pollution score was only used at the shoreline. This study is using it on a coral reef, and it shows that, yes, you can use it on a coral reef as well. The sewage pollution score is also important to identify areas of concern so that we can focus our efforts on these areas and address the impacts to these areas. This study also raises awareness of sewage impacts to reefs in Hawaii, both Hawaii State and Hawaii Island. It shows that sewage pollution can impair coral health, and it also emphasizes that sewage pollution increases the vulnerability to other stressors. In this case, all of these impaired health conditions are likely due to the stress imposed by sewage. And finally, the study also illustrates the need for improved sewage treatment and disposal in coastal communities. It shows that sewage can impact offshore reefs, and that we do need to find some solutions that are not only environmentally friendly, but can also fit the needs of our communities and those who live near the ocean. So as I found out in my thesis research, doing 20 stations throughout the South Kohala coastline is a pretty big effort. So there's a long list of people that I'd like to thank. I'd like to thank my thesis committee for guiding me and helping me along the way. The Puako Community Association, thank you for providing us logistical support and really being supportive of the project. I'd like to thank the boat captains for putting in those early mornings and long hours on sea. My family members for the support. The Puako research team, thank you for helping me along the way, answering my questions and lending me some gear as well. My pipes interns, thank you for waking up early and putting up for the long days on the water. Thank you also to the research divers who helped me collect my data. Thank you to the Analytical Lab for processing our samples. Thank you to these individuals for their help making Clod cards and lending me equipment. And also thank you to the funding sources. This project wouldn't be possible without you, as well as my graduate education, especially. Thank you very much. Thank you all for listening today, and I'll be free to take any questions you have. Thank you. I see Craig's hand up.
Nice work, Devon. That was great.
Thank
So you have this really interesting pattern with the area up around the Mauna Kea resort. And you, I think, offhandedly mentioned, and apologies if I should know this kind of stuff from past work, but you kind of offhandedly mentioned that this was a little bit of a new observation, and you know, given the context that you're able to provide with the past work done sort of more south along the Puako coastline, and the fact that there's a really high sort of abundance of turf algae up there along the Mauna Kea. Is there any historical data on what that place was like maybe 20 years before or 30, I don't know exactly when those resorts were put in, and whether the benthic cover has changed, which might be an indicator that, you know, sort of that temporal perspective. I don't know if anybody knows anything like that.
Let's see. So to answer your question, I guess from a literature aspect, I don't know if there's been any studies aside from like an EIA, EIS done there.
Yeah.
I know the Nature Conservancy does have at least one monitoring station there. I could kind of look and see what they have and how far back their data goes, but more qualitatively, the surveying there. Actually, I should put this into perspective. In around, I think, late 2015 to early 2016, there was a big bleaching event that happened along the entire leeward coast of Hawaiʻi Island. And I know the South Kohala region was one of the hardest hit. And just doing surveys there and collecting water samples, swimming around there, you could see there used to be these really big tracts of coral reef that are now just covered in turf algae that you could tell used to be, you know, healthy and at least living. So my guess is that some of these other kind of bigger stressors, such as increasing ocean temperatures, coral bleaching, they're also kind of driving that change, that benthic change in the North Resorts region.
Great, thanks.
Yeah, thank you.
Hey, Devon, you have a question from Ron Kittle. He says,
oh, okay.
do you think there will be a difference with turf algae if you delimited among the major taxa? Turf can be red, green, brown, and cyanos with different mixed assemblages. Are they similar assemblages in all locations sampled, in values of nitrogen or whatever isotopes, etc.?
Okay, good question. So I don't have it on my PowerPoint here, but I do actually have a list of the algae species we collected to run N15 samples. As far as the actual turf algae, we didn't try to identify those two species. From what I understand, it's currently really difficult to do that. And it was also hard to actually collect turf algae samples. However, just kind of qualitatively, what I've noticed, there's a lot of cyanobacteria on the reef. Most of it seems to be in the 3 Puako regions. There appears to be more actual like turf algae involved in algal overgrowth in the South Resorts and North Resorts regions, but some Corallophilia, Huismansii, and some other kind of brown colored turf algae. That's what I have noticed. Like I could do a little bit of digging and kind of link the coral species to their N15, not the coral species, the algae species to their N15 values as well. So I think that's something kind of worth looking into and seeing if there is some sort of association or trend with that.
Any other questions for Devon?
So Devon, if you were to move this project forward, what do you think are the most important next steps for this region for better understanding the impact of sewage pollution in this area?
Personally, I would look into the North Resorts region some more. That really surprised me. Part of it might be that we only had 3 stations there, and there is kind of a limited amount of coral cover there, so we really could only find 3 suitable spots to do this. But I'd really like to look in that North Resorts region some more. I'd also be, I know, not to appease to your oceanology side, but I'm really curious too, looking at the whole coastline, how looking at water motion and circulation at a larger scale, kind of seeing if, you know, if water maybe from a more polluted region, say the South Puoko region, is maybe moving up and is being caught in that North Resorts region, or something similar to that. Maybe looking at a larger scale, how water motion just works along this coastline.
Hey, Devon, why don't you share with the audience what you were telling me about the North Resorts watershed? What else was there just besides the resorts?
Oh, right. So another point about the North Resorts region, in addition to the luxury resorts there are several hundred private residences located upslope. And one of the kind of side projects I looked at was looking at the locations of OSDS units from state planning data just in the South Puoko or South Kohala region. And it was interesting to note there were, in the location of these private residences, there are about maybe 20 to 30 OSDS units. And this is kind of surprising because the resorts in the North Resorts region use sewage treatment and actually use the treated wastewater to irrigate. So this is kind of surprising. I mean, I kind of want to almost ground truth it in a way, you know, maybe contact the resorts or see if these private residences are connected to the sewage treatment, or if they're not, or what's going on there.
Hey, Devon, you have 2 more sets of questions. So the first one is from Corey Anger. In your reading, have you found examples of successful methods to implement to remedy the negative impacts on the reef due to sewage?
One of the things I've been reading is making a smaller scale sewage treatment. So here in Hawaii and also the US to some extent, it's kind of a trend of large scale sewage treatment plants where an entire, let's say an entire portion of the island is serviced by 1 or 2 wastewater treatment plants that receive that wastewater from various communities and developments along the way. And reading into this, one of the things that is commonly or has been increasingly employed in Europe is the use of small scale sewage treatment. So each town or each kind of settlement will get its own sewage treatment. And even from there, and especially in West Hawaii, reusing treated wastewater, they have the capability nowadays to really treat sewage and make it, you know, to how should I word this, to make it more appealing in a way so that you can use it for irrigation or agriculture.
You can even drink it in some cases.
Or even if you invest the time and money into it, you can even drink it. So something like that, especially in, you know, dry and arid West Hawaii, I think it could definitely be an asset for sure.
You have one more, got a couple questions here. So the next one is from Matt Connolly. I'm wondering if anyone is considering doing any sampling now that the resorts are operating at a much lower capacity due to COVID-19 and looking for Delta 15N signals in the groundwater, etc. Yes,
that's something I was thinking about in the last few days, especially even with less people in the water. That I'd definitely be interested in that, just to maybe see if there's any association with the population of people or even density of people in that area.
Next question is from Lindsay. Hers is algal turf colonization is very common on portions of coral colonies with partial mortality. And algal overgrowth can sometimes be difficult to score as algal turf is almost always present along the edges of live coral. Was there a particular threshold of overgrowth you used to score algal overgrowth? Second, were there particular species that were commonly observed overgrowing corals? And three, is there an understanding of coral health condition prevalent in adjacent areas with low sewage pollution?
Okay, so the first question was pertaining to how we basically determining if algal overgrowth was occurring or not. And to do that, we did our best to really get kind of up close and personal and look to see if the turf algae was actually growing in the living coral tissue. And I did some, with all the divers involved, we, with the help of Dr. Courtney Couch, we did little like training sessions just to make sure we were all calibrated and seeing the same thing. And the data generally reflected that because algal overgrowth happens at these thin little margins, most of the time it was, you know, like 1 to maybe 3% algal overgrowth occurring on an individual colony. So I feel pretty good about how the data turned out and everything as well. The second and third question, let's see. To address those, I would be interested in comparing this data to an area that isn't as sewage impacted. I'm thinking it might have to go down to like South Point or someplace pretty remote, just because I don't have a figure. Well, I can actually kind of go back to the Hawai'i Island map, but even like most of the Kailua Kona area has a high OSDS density, so I really have to find some remote spot or find a study that did it in some remote spot. But it would be definitely interesting to look at that and see if there is a difference even in the algae species or percent cover. I think the second question was asking about the algae species or the coral species involved. The most common coral we encountered during surveys was Parietes lobata. There was occasionally some Parietes meandrina, some Parietes evermanni, and maybe something kind of unique and rare like a Fungia scutaria. But for the most part, it was Parietes that were being affected by these impaired health conditions. And from the most part, it was either red or brown turf algae. If there was any kind of green algae, it was like a greenish cyanobacteria that was growing on the coral, but that wasn't as common as the brown or red turf algae.
Any other questions for Devon? All right, thank you, Devon, for giving us such a great presentation. And everyone, let's do a round of applause. All right, so our next step is we'll be ending the public presentation, and then Devon will be meeting with his committee to talk more about his thesis. So thank you, everybody, for joining us, and have a good rest of the day.
Thank you, everyone.
Devon, I don't know if you can see the chat stuff, but if you click on.
Okay, there it is. Oh.
Yeah.
Oh, wow, thank you.